Thank you. All right, so today we're going to be doing normalization prior to peak calling, where we're going to be looking for significantly enriched regions in our chip as compared to our control, which is the inputs. And then we will be doing Genome Browser viewing using the integrated Genome Browser program, and differential peak calling using a program called Homer. So we ended yesterday with our, we downloaded FASTQ files, then we did bowtie 2 alignment, and we generated SAM files, which are files that have alignment information for our sequence reads. So with those, we will do max peak calling and then generate bed files, bed graph files, wiggle files, and bigwig files. And I know that maybe some of you ran into trouble yesterday and weren't able to generate SAM files. So I made the SAM files in a compressed folder that you are able to copy from my folder into yours. So if you need to get the SAM files, you weren't able to generate them yesterday, you want to participate today, you can use these commands. Where first you'll have to log into Hoffman and then go to your Flash Scratch folder. This is if you created the Flash Scratch shortcut, which you can just do CD Flash Scratch from your home directory, copy the SAM files compressed folder, then decompress that, go into that folder, and then move the SAM files back into your Flash Scratch. This should be a lowercase c. Sorry about that. But I'll copy these into the chat right now so we can proceed to the next window. So 1 second. So again, this is for people who weren't able to generate the SAM files. They got stuck on a step yesterday. So those are the commands. Again, this is after you log into the Hoffman server. So for the rest of us, hopefully you have your SAM files, and we can proceed with normalizing the SAM files. So what we want to do is get roughly equal sequencing depth between our CHIP files and our input files. That way we can have valid comparisons when we compare the 30-minute interferon alpha treated chip, STAT1 chip, and the 6-hour interferon alpha treated STAT1 chip. So we want to compare those two files, but in order to do that, we have to normalize for equal sequencing depth. So we will request an interactive session. I'm sorry, I don't, must have left that out. So let's start by logging in. I'm gonna go back to here. So this is how we'll begin. So on page 34, again, we're gonna log into Hoffman, we already created a shortcut, and then we're gonna request an interactive session with, instead of the 2 hours, change this to 3 hours. So I'll demonstrate that right now. Terminal. So we are going to log in. Again, use your username to do it. Your password. Now we are going to request an interactive session. the following command. And again, if you want to reference the slides, this is on slide 34. Except instead of 2 hours, we're going to request 3 hours because we're starting earlier today. And again, I'm requesting 4 nodes. So this might take another minute to be granted. So we'll have to wait for that. So once that goes through, we will enter the flash scratch folder with cd flash scratch, and then we are going to load the program SAM tools. So this is a program that comes already installed on Hoffman cluster, but in order to use the program, you have to load it. So we'll use the program or the command module load the same way we loaded Bowtie 2 yesterday. And then, so we're gonna write a custom shell script to, first we're going to filter out mitochondrial and unassigned reads with a sed command. So we're gonna remove the reads that align to CHRM or chromosome M for mitochondrial chromosome, and then the reads that were assigned to random or unaligned. So these are in the reference genome as sequences that they know exist in the human genome, but not exactly sure where they exist. So we will remove those because they're not going to be important to us, and it can actually complicate the statistical analysis from the peak calling. So by using this command, we will filter out those reads, and then we're going to convert our SAM files into BAM files. And SAM files and BAM files are related, but the difference is BAM files are compressed into a binary format. So they are smaller format and easier to use because they are smaller files. And then we're going to use SAM tools again with SAM tools view -C option to count the reads that are in our BAM files. Then we'll use these numbers to normalize for equal reads. And what we're going to do is downsample the file that has more reads to match the number of reads in the sample with the lower amount of reads. So it's not important to have the same amount of reads in the input and the IP, since peak calling will account for that. But if we want to compare between 2 different IPs or 2 different chips, we want to have equal sequence depth, equal genome coverage. Otherwise, you can imagine if you have twice as many mapped reads for one, the peaks could be twice as high, and it looks like there's increased enrichment for binding in that sample. So I'm going to go back to terminal. Hopefully the interactive session was granted. It was. So now I'm going to enter the flash scratch folder. I'm going to load SAM tools. Now I'm going to create a simple shell script with these 3 commands, so that way I'm not having to do these 3 commands each time for all of the files. I mean, of course there's many different ways to do this. This is just a simple way that I thought would allow us to get through this quicker and relatively simple. So as you can see, I already made this bam.sh shell script. So what you're going to do is when you hit nano bam.sh, for you it'll show an empty page like this. And then you can type in these 3 commands, or you can copy and paste them in. I'm going to copy and paste them to save time. And then if you hit Control X to exit, it'll ask you if you want to save the modified file or save modified buffer. Hit Y for yes. And then it asks if you want to call the file name what you put after the nano command, which is bam.sh, and we do, so we'll hit enter. Now we created that simple shell script, but in order to execute the shell script, we have to change the permissions. So that's on the next page here with the chmod 755. This is a numerical code that'll change the permissions. It'll modify the permissions of the bam.sh. So this will allow the file to be executable. By default, it won't be. So I'm going to go back to terminal. chmod 755 bam.sh. Right, so now I can execute this file, and what I want to do is tell it that to execute it, you want to do // so it's this directory, this file, and then what we did is we added a $1, which is a variable, which says for the SAM file that begins with this prior to the .sam extension, input this variable into the command. So going back to the command, just to explain this a little better, what it's going to do is it's going to take the stat1 30m interferon alpha .sam file. So it's going to replace the $1 with that variable we put after the execution. It's going to output a filtered version of the SAM file where we remove the mitochondrial reads and the unassigned reads. So that'll output a file that is stat1 30m ifna filtered.sam. And then we're going to convert that filtered SAM file into a BAM file. And then it'll count the reads we have in that BAM file. So I hope that makes sense. And it probably is still running, but I don't think it'll take too long. So you'll know it's finished when it'll add a number at the bottom, which is reporting the number of line reads we have. And again, we're doing this so we can normalize for equal mapped reads. There are probably, well, I know there are programs that do this, but I'm just going to do it with this simple shell script because it's not a very complicated process. Yeah, okay, So I had a good question. So the -c parameter means count. So we're using SAMTools view, the -c, to count the lines in the BAM file. The -b means output as a BAM file. So we're converting the SAM to a BAM with that parameter. So if it gave you an error that says truncated file, that seems as though there's an error during the alignment process. So if that's the case, I recommend using those commands I pasted above to retrieve the SAM files that I have in my folder and use those files, because it seems like there was an error when doing the alignment yesterday. So you can see this finished. We have 11.3 million reads in this file. So now I'm going to do it again with the 6-hour SAM file. So to leave the text editor, it's Control X. And then it'll ask you if you want to save the file. You hit Y for yes or N for no. And then it'll ask you if you want to save the file as the particular name that we put, and then hit enter. All right, so the second one finished, and there's 11.4 million reads. Next, we're gonna do the same with the input files. Okay, so after the last one's finished, what we're gonna do is we want to get the same number of reads between our ChiP samples and between our input samples. So an easy way to do that, it's a rough way to do it is using SAMTools view. The -b means input BAM file. The -s means sample reads. So what we're going to do is we're going to leave the two ChiP samples alone because they're already very close to the same number of reads, 11.3 million versus 11.4, which, you know, a 1% difference, less than 1% difference, not going to change our results in any way. So I'm not gonna change those files. And what I guess also what I noticed is that my samples are a little different than when I did this. So I don't know. I noticed that I get a slightly inconsistent number of reads. I think maybe from the connection being unstable between Hoffman and NCBI where we downloaded it. But what we are going to do, mostly just to practice, is to subsample input reads. So the input reads, we have about 19.5 million and over 20 million. So those are also pretty close, but we could get them closer. But what we're also going to do is not because I would normally do this, but we're going to sample these down to about 1 million mapped reads, just because it'll help make our processes that we are going to be running later with these files. It'll make them go faster. Instead of having 20 million, we'll have about 11 million. So what we're going to do is we're going to, for the input 30-minute file, dividing by 11 million by the 19.5 million, it's about 0.564. So we use that number in the SAMTools view command, -s. And if after the minus lowercase s, you do 1 point and then that fraction, that number when we divide. So it's going to multiply the number of reads by 0.564 and subsample that number of reads. So I don't know why it wants to start with a 1, but you have to do that. And then we're going to output that input into another BAM file with roughly 11 million reads. And then we'll do the same for the other input, 6 hours. And the reason I chose 11 million is just because I didn't want to have less reads than the IP, but the 11 million is because I want to have less reads just for the purposes of this workshop, just in the interest of time. It'll speed up analyses we're going to be doing downstream. But normally I wouldn't throw away that many reads when I want to keep as many as I can. But just for the interest of time for this workshop, we're going to sample down. But what I would do is I would want to sample the same number of reads. So I would downsample this 24 to 19.5 million. So this is going to give us roughly the same amount of reads for the inputs when we do this. So I'll demonstrate that in terminal. Don't worry if your numbers are slightly different than mine. I've noticed that there's slight inconsistencies when obtaining data from NCBI to Hoffman through the SRA toolkit, but they should be close to these numbers. So again, I'm sampling 0.564 times the reads from the input 30-minute interferon alpha to make a smaller input to obtain roughly 11 million reads for both of the inputs. And this will allow our peak calling to go by a little faster. Yeah, so again, for some reason, the version of SAM tools that we're using, you have to put 1 before the 0.564, and I'm not clear on why that is, but that's just what the parameter calls for. I remember looking it up at one point, but I don't remember what it was. I know a different version of SAM tools, you enter the fraction in a different way. So that one finished. So now I'm going to go with the 6-hour input. Oh, whoops. I messed up because I used the same value, but to stop it, I hit Control Z, and I want to change this one to 0.538 because there's a slightly different number of reads. Okay. So now, if I wanted to, I could count the lines in this BAM file by using the SAM tools view minus C option of my output input. and it should, if it worked, it should give me around 11 million reads. It seems like maybe a couple people are stuck. Yeah, so 11 million. So I'll try to make it so these BAM files I just made are available to copy into your folder. So give me 1 second. So to stop command, sometimes it's Control Z or Control X or Control C, and on PCs versus Mac, on Mac, usually in terminal, it's Control Z to stop a command. And PC, I think it's more typically Control C. I don't use a PC, so I'm not positive on that. Different programs use different. Sometimes a program will require Control X or Control Z or Control C. I'm not really sure why or on why. I just try one. If it doesn't work, I try the other. So if you're having trouble generating BAM files, I will give you a command. I'll paste it into the chat room so you can copy the BAM files to your folder. So it'll just be 4 commands to copy each one. Let me know if it doesn't work. I did something wrong. All right. So now that we have normalized BAM files, we're ready to. So a question: how to see the count number in the BAM files? It's. Let me make sure I'm sharing the PowerPoint. So you can just use the command we put into the shell script, the last one, the SAM tools view -c, and then the name of the BAM file you want to count. So it would be the SAM tools view -c, and then the input, for example, with the 11 million.bam, that it'll report the number. All right, so the peak calling program that we're going to use is Max. Actually, we're going to use Max version 2. It's a slightly updated version. So what Max does is it models the length of the chip fragments, which is what we talked about yesterday, with the short reads being on either sides of the fragments. It'll find the distribution of those reads, and then use modeling to determine the fragment length. I'll get more into that in the next slide. The tag distribution along the genome is modeled by a Poisson distribution. So after it determines the fragment length, it models it with a Poisson distribution, and it empirically will estimate FDR for each detected peak over control. So for us, it's going to use our input file and use local windows of different sizes in the peak region that it wants to determine significance for. So it'll estimate the expected number of reads in that region using these, what it determines is a lambda value for by modeling the or by counting in different window sizes and the peak range for the control. So again, the modeling portion of this allows Max to more accurately predict the true binding site by taking the distance between the pileup of reads on the Watson strand, for example, and the distance between the peak of the Crick strand or the other strand. So the distance or the fragment length is estimated by the distance between these two peaks. So theoretically, the reads on this end of the fragment is a distance d, the length of the fragment to this, the peak on the other strand. So then it will shift the tags by this distance divided by 2. So halfway in between this distance, that's where the tags will be shifted. So you have a theoretically more accurate positioning of where the binding site is for your peak. Of course, if you have paired end reads, which are becoming more cost effective these days with the technology that's developing with sequencing, it'll ignore this modeling. Because you don't need to model, the paired ends will determine the true size of the fragments. But we are using single end today, and single end is really sufficient for ChiP-Seq. Paired end is not necessary, but being that it's cost effective today, it's becoming more popular. So in order to run Max2, we have to install it. It doesn't become, it's not pre-installed like SAM tools or Bowtie, but it's easy to install with this command, pip install, which is the command that can install a lot of different Python programs easily. So first we have to load Python, and this is the version we're gonna load. I think this was, I don't think it's the default. So if we just did module load Python, it'll load the default version of Python, but I think this, the reason I chose this version is because I think it's, I know it's compatible with Max. And then we're gonna install Max2 to our user directory. So we can't just install any program we want to the Hoffman cluster, but we can install programs to our local user directory. So when we call Max2, you have to use your user directory in order to call it. Instead of, if we just typed in Max2, it wouldn't be recognized. But if we put the path to the Max2 where it was installed in our user directory, which is here, again, you need to replace the this with the first letter of your username and your username, and we're going to be using Max2 call peak algorithm. And if this was installed correctly, when you type in this command, you should see this pop up. So I'll go ahead and do that. Okay. So for me, it's going to say, it might give you a couple errors, but don't worry about that. That's okay. For me, it says the module's already loaded. Then the pip install command to user directory. And for me, I already installed it, so it probably shows something different for you. So if after we close terminal and you log back in and you want to run Max, you don't have to reinstall it, but you will have to load Python again. So to call Max2, I'm going to direct it to my home directory where it was installed, which has this location. And then Max2 call peak. It should give you that. Hit at the bottom, I'll say there's an error because we didn't tell it what files we wanted, but we were just pulling up the options here. Okay, so now that we installed Max2, we can run it. So I know that I stretched this out so it'll fit, but it's kind of small, but this is the command we're going to use. -T designates the treatment file, which is our CHIP file. -C is going to be the control, which is the input. -N is the name we want to give to the output files. So this is going to be, first we're going to start with the stat1 6-hour interferon alpha. -G is the genome size. So if you put HS for Homo sapiens, it already has that number predetermined. But if it was a genome where it didn't, not uncommon genome, for example, you would put in a value for the genome size. The -BDG means output a bed graph file. So that's a file we can use for downstream programs. -Q, so this is the value, Q value of less than 0.05 will be required for us to call the peak a significant peak. So that's fairly stringent. You can use P values. I like to be more on the stringent side and use the Q value because I would rather eliminate false positives. And then -F is the format of the input. So we're inputting BAM files. So I think what we'll do, because this might take around 20 minutes to do each one. So what we'll do is we'll start the first one. When that finishes, we will jump ahead and make some files that we can use for the Genome Browser, and then we'll download the Genome Browser to our actual computers. We can't open the Genome Browser from the Hoffman cluster because there's no graphical user interface. And then while we're playing around with the Genome Browser, we'll run the second peak calling. So we can do that now. And if it worked, you should see something similar to this, where it'll tell you the files you input, the genome size it's using. We're using modeling, the Q value cutoff, at paired end is off, etc. And then it's going through each read tag, and it reports 1 million at a time. So it'll go through to 1 million for each, I think for both the chip and the input. And then when that's done, it'll generate the bed graph file, which is like a pile up of the reads for visualizing the chip signal. And then it'll determine significant peaks. It'll do the peak calling portion. So again, this might take about 20 minutes. So this is a good time to stretch your legs, ask questions, give your cat a haircut, whatever you want to do. I'll stick around and answer any questions if you guys have any. So while the peak calling is happening, I'll describe the output files. So what Max generates, we'll get an Excel file with information on the peaks. It'll report the statistical value it used, the peak location, the position of the peak summit, so the highest portion of the peak, and it'll also output a bed file. So this is a more simplified version of the peaks file, which is a specific format that can be used in other programs, but it's essentially a list of the peaks. And then a summits.bed, so instead of the whole peak width, it's just the coordinates of the summit of the peaks. So this is great for doing like transcription factor motif analysis. So you can pinpoint or estimate more accurately the true binding site if you just use the very summit of the peak, because with CHIP the resolution is not very fine. Negative peaks is a portion where you have de-enrichment compared to the control. I can't think of a use for that, but they provide it. An R script, which can generate the modeling graph that I showed earlier, showing the modeling it used for the determining the fragment length by looking at the distribution between strands. So we're not, this is Max, but for Max 2, it doesn't, instead of a WIG file, it outputs a bed graph file. So we're going to get a bed graph file and a diagnosis report. And if we put in the parameter for sub peaks, so you can also determine smaller peaks within peak regions, which are called sub peaks, but we didn't use that parameter. It uses what's called a peak splitter. So split peaks into smaller sub peaks, which we didn't use. Also, if you're chipping something that you're expecting enrichment in broad regions instead of sharp peaks, you can use the broad parameter, -- broad. So things like heterochromatin marks, like H3K27 trimethylation, you expect it to be enriched in broad regions. So I think it does a better job. I've never used it, but I think the -- broad will probably do a more accurate job at calling these peak regions or these enriched regions. Okay, so when that finishes, like I said, instead of, we'll come back to the second chip. So we can have that running while we are looking at the 6-hour chip in the Genome Browser to speed things up a little. So what we're going to do, where is it? So what we're going to do is, first we're going to convert the bed graph output into a wiggle file, not because we're going to use the wiggle file right now, but later when we look at average signal using some other programs, they require a wiggle file. So we're going to convert the bed graph to a wiggle file. So I just put into Google bed graph to wiggle conversion. I found that somebody's script that they uploaded, a Perl script. So we're just going to download that script. It's really simple. We will use it to convert the bed graph into a wiggle file. And we're going to use a step of -50. So that'll help to reduce the file size. So instead of a resolution of binning the genome by a single base pair showing a Y value or how many reads were aligning to that individual base pair, we'll bin the genome into 50 base pair regions. So that'll help to minimize the file size. It'll shrink the resolution of the wiggle file, but its CHIP resolution isn't smaller than 50 base pairs anyway, so it's fine to do that. And we're also going to convert the bed graph into a big wig file. So this is, again, similar to a wiggle file, but it's a binary format, so it's a more compressed and smaller file size. And this allows Genome Browser viewing to be quicker. So if you have a big wig file, which is smaller than a bed graph file and smaller than a wiggle file, it's easier to load. It'll load quicker in the Genome Browser than, say, a bed graph or a wiggle file. So to do this, we'll use a script from the UCSC suite of scripts. It's called bedgraph to bigwig. We'll download that. But we also need a file that has the chromosome sizes of the HG19 index file that we're using. So we'll download a script to retrieve the chromosome sizes. And that script is called fetch chrome sizes. And then we have to change the permissions to execute these two scripts, again with the chmod command. And then we will retrieve a file for the chromosome sizes of HG19, and then we'll run the bedgraph to bigwig script to convert our bedgraph to bigwig. And the reason we need the HG19 chromosome sizes is that the bedgraph to bigwig script requires that file to convert the bedgraph to bigwig. And once we do that, we will copy these files to our own computer. So we have to transfer them to our local machine that we're on, not on the Hoffman server, in order to visualize them in a Genome Browser. And we're going to have bigwig files for both the chip and the input. So we can compare the enrichment between the chip and the input. So we can visualize how the peaks differ from the control. So we can see if we agree with the sites that were called it for peaks. Also, we're going to take one of the bed files and convert that, sorry, one of the peak files. So we're going to convert that into a more simpler bed file, which will also transfer to our computers, and we can load that into the Genome Browser as well. So we'll use this simple cut command to reformat this output peak file. The peak calling's still going, so I'm gonna wait for that to finish. So when you guys do this on your own, when you generate your own chip data, you can, of course, open up multiple logins for the Hoffman, and you don't have to wait for one to finish necessarily. You can request multiple interactive shells. And also, something we're not doing, but you probably learned in a different collaboratory course, is you can submit jobs into the queue where you aren't having to, where the jobs are running basically in the background on the Hoffman, and you don't have to stay logged in and sit and watch them run. And you can run a lot of parallel jobs that way as well. So as soon as this finishes, I will start the conversion of the bed graph to a wiggle and the bed graph to a big wig, and also the peaks file into a bed file. And then I'm going to download the IGB, the Integrated Genome Browser program. So of course there's other genome browsers software. If you're familiar with other ones and you have one that you like better, feel free to use that. I like IGB, so I'm going to be using that one. Okay, so my peak calling just finished. If I want to look at the files, so I have the R file for generating a modeling figure. So just a warning, if you do that, it's not going to look good. And there's a couple reasons why that is. This is not the highest quality of data, and we're not using the optimal number of reads for transcription factor. And also, there's not a lot of STAT1 binding sites on the genome. I think that is true biologically. It's mostly just activating genes involved in protecting the cells from infection. So we have a bed graph file. The .bdg extension is the bed graph. So we have it for our treatment file, which is our CHIP. We have it for our control, lambda, which lambda is the control. This is our input. Bed graph file is generated from the input. We have an Excel file with information on the peaks. We have a narrow peaks file, and then we have a summits.bed file. So again, the summits is a more narrow version of the peaks that were called. So what we're going to do next is what's on slides 54. We're going to download this Perl script to convert the bed graph to a wiggle file. So I'll do that now. Kind of a long extension, so I'm going to copy and paste it. Yes. And then we're going to change the permissions of that script so we can run it. I think there was an error in the naming of the file, like there was a space before the name, so it started with a %20, but in any case, doesn't matter. chmod 77755 %20 bedgraph to wig.pl. You can rename it if you want, but I'm just going to keep it like that. So now I can execute this script by typing Perl, the name of the script. Our bedgraph file that we're going to convert is going to be stat1 6-hour ifna treat pileup.bdg. Then our output is going to be a wig file that I'm just going to call stat1 6-hour ifna.wig. And then the step size I'm going to use to reduce the resolution in the file size is a step of 50. So this is every 50 base pairs, you'll only have one Y value. That will become clearer when we look at it on the Genome Browser. I don't think this should take too long, maybe a few minutes. And again, the reason we're doing this is because some of the programs we're going to use downstream of the peak calling require a wiggle file. They're, it's a similar file to bedgraph, but for whatever reason, I think a wiggle file was is a more outdated version of the bedgraph, and maybe those programs are a little older. But we'll be using this wiggle file, I think, tomorrow to visualize the average signal over genomic regions of interest. But the bed graph, the wiggle file, and the bigwig file can all be loaded into a genome browser to visualize the data. But what's recommended is using the bigwig file for that, especially if you have a lot of data, you have a lot of reads. The file size is going to be a lot smaller for a bigwig than a bed graph or a wiggle file. So it'll allow for easier viewing in the genome browser. Oh, and I realized that I don't think I'm sharing the screen I meant to share. Okay. I meant to share this, but yeah. So what I did is I typed in the commands on slide 54 to download the bedgraph to wig Perl script. I changed the permissions so we could execute that script. And then I used the Perl command. Now I'm converting the stat1 6-hour CHIP bedgraph file to a wiggle file. All right, so the CHIP file finished converting. So I'm going to do the same for the input. So instead of stat1, I'm calling it input 6-hour interferon alpha.wig. And then I need to change the bed graph to stat1 6-hour -a control lambda beg. So again, I only ran the script, the peak calling script on the 6-hour stat1 file. So we could use the Genome Browser and we could generate the files for the Genome Browser and then load them into the Genome Browser and play with those files while the other peak callings, the peak calling for the 30-minute interferon alpha treatment was running, just so we're not sitting here doing nothing for another 20 minutes. It'll allow this to go by a little quicker. So I'm gonna, after after I make this, and then I make the bed graph, and then I make the bed file, then I will transfer those files to my computer, my actual computer's hard drive. I will download the IGB Genome Browser and load those into those files into the Genome Browser, and then while that's happening, I will run the other max2 peak calling program. All right, great. So the input finished, generating the wiggle file. So the next thing I'm going to do is convert the bed graph into a bigwig file. And again, this is the file type that's preferred for Genome Browser viewing. So we're going to download a script from UCSC, the bed graph to bigwig script. And we're also going to download a fetch chromosome sizes script. So this will get a file that is required for the bedgraph to bigwig script. So what I'm going to do is use these commands to download these scripts, change the permissions so we can execute those scripts, fetch the HG19 chromosome sizes file, and then convert the bed graphs to bigwig files. So do that now in terminal. It's a pretty long URL, so I'm just going to copy and paste it. Okay. The fetch chromosome sizes file, I already had it in my folder. That's why it says 0.1, but I wouldn't say that for you. And then I need to change the permissions for bedgraph to bigwig. chmod 755 will make it executable. Now with the fetch chrome sizes file. Okay, now I'm going to execute fetch chrome sizes, retrieve the chromosome sizes of our HG19 reference that we've been using. So you want to use the same reference that you align to. And output it as a file called hg19.chrome.sizes. Okay, so if I look at that file, this is what it will show me. It's just a pretty simple file. It says CHR1, and then it has the number of base pairs. So it's just 2 columns. All right. Next, I'm going to use the bed graph to bigwig. So our bed graph file is stat1 6-hour IFNA treat pileup.bdg. And again, it needs this hg19.chrome.sizes file. And then you put in the name of the bigwig file that you're going to generate. Stat1 6-hour IFNA.bigwig. You run that, and it should be generating our bigwig file. Oh, that finished. It's a lot quicker than the bed graph to wig. So now I need to do it again with the input. So I just pressed up to retrieve the last command I entered. I'm editing the text to input. And then instead of inputting the CHIP file, I want to input the input bed graph. Control and PDG. And if you're confused about what we're doing exactly, it'll make more sense once we start using these files, and you can understand why we're generating them. Right now, we are setting up to use downstream analysis that will actually give us data by doing the kind of boring stuff now to generate all the files that are going to be necessary to hopefully get interesting data. Okay, so that finished. We will next convert our peak file into a bed file. So again, this will be a file that we can load into Genome Browser, and also we'll be using it for a few other programs, probably tomorrow, I think. Basically, this is going to be a very simple file of just the locations of the peaks, really not any more information than that. To generate it, we're going to just do some editing of one of the outputs from Max by cutting some of the columns out of that file. So that way it'll be compatible with all of the different programs we want to use it for tomorrow. So this should be very quick. Just cut F1, 2, 3, 4, stat 1, 6 hour, IFN, A, peaks dot narrow, and output it to stat 1, 6 hour, IFN, A, peaks dot bed. Okay, and that's it. So now if we were to look at this file using less command, this is what it would look like. So basically it's just the chromosome, the position of the peaks from start to end on that chromosome, and then some unique name that was given to that peak. To get out of this, you hit Control Z. So in the file that we used to generate this, like this, so we remove some of these lines. So I can't remember what all these columns mean, but if we opened up the Excel file, I don't know if it'll look right on this, but the Excel file will have more information about what each column means. What is the name of that? Yeah, so again, it has the chromosome positions, the length of the peak. I think this is the summit location. These get a little goofy. The pile up of reads, I think it's this column, yeah. The minus log 10 p-value, the fold enrichment, the negative log 10 q-value, and then the unique identifier. So if you want to know more information about each peak, I recommend opening up the Excel file that outputted. So now, before we run more of the peak calling, what I'm going to do is copy these files to my computer so we can take a look at them on the Genome Browser. Well, I guess before doing that, we are going to download the Genome Browser. by going to the website. So let me open that here. So this is that link that I put into at the bottom of slide 57. So right here, all you have to do is go to the top right corner, hit download now. Download that, it'll save the file. It's not too large, but it'll take half a minute or something to download. I'm gonna open that up. And I don't know if you can see it, but I'm installing the Genome IGB right now. Which might take a minute. Let's see. Then I'm going to hit run. It's not the fastest program. It takes a little while to open. So if it's taking a few minutes, don't worry. It's just going slow. So while that's opening, I'm going to go back to PowerPoint, and I'm going to copy the files that I want to load into the Genome Browser. So you can use these commands I have on slide 59. What they show is just making a folder on your desktop to make it easy with the mk make directory command. I call it ChiP-Seq workshop. Of course, this is not going to be on a login on your Hoffman server. You'll have to open up a new terminal tab or a new terminal window and then navigate to the desktop. On a Mac, these will be the commands. It might be a little different on a PC. And then I'm going to use a secure copy command to copy them from my username. Of course, you would have to change the red text here if to the location of the file. So this is going to be the bed file, the bigwig file for both the chip and the input. And then space, and then this is the folder that I'm going to copy them into. So I'll demonstrate that. Of course, you can use FileZilla if you prefer, or CyberDuck if you have experience with that. But I'll demonstrate how to avoid using those just with this secure copy command. So again, I'm going to open up a new tab in Terminal with Command T. It might be a little different if you're using Cygwin or a different PC Unix-based command program. And I'm going to go to my desktop. I'm going to make a directory that I call ChiP-Seq workshop. Of course, you can call it whatever you want. And then I would hit enter, but I already have this folder, so I'm not going to do that. And then this command scp, secure copy, the location of the file. So you have to tell it your username and then the Hoffman address. Similar if you were using FileZilla. So it's going to be after the colon, you do your home directory, flash scratch, and then the file is called stat1 6-hour ifna peaks.bed. Then I want to copy it to this directory I just made, the ChiP-Seq _ workshop. So it's just a space and then where you want to copy the file to. I'm going to ask for my password and then the file transfer it over very quickly. This bed file is really small. And then I'm going to do the same thing for the two bigwig files. These are a little bigger, so it won't just be instant transfer, but it should only take 10 seconds or so, depending on your how fast your internet connection is. So now I'm going to do it with the input file. And just in case we need a refresh on FileZilla, if you prefer to use FileZilla instead of the SCP command, it's a little more, it's more visual, so it might be more helpful. So for the host, it's hoffman2.idre.ucla.edu, same as when we log in. Our username, of course, is our username. And the password, the same password we use when we log in. And oh, you guys aren't seeing that, sorry. So FileZilla, hopefully this is now visible. Host is the hoffman2.idre.ucla.edu. Username is Zemke. Password we use to log in. And then the port is 22. Hit quick connect. Now I'm connected. I want to go to Flash Scratch. And then I have the files that we wanted somewhere in here, somewhere in this mess of files. The two bigwigs and the bed, I would just copy them over to whatever location I wanted. So for me, it's going to be the desktop and then the ChiP-Seq workshop folder I have. And just copy and drag them over. I already have these files in that folder, so it's asking me if I want to overwrite them. I'm just going to hit cancel, cancel, cancel. All right, so that's if you want to use FileZilla. The important part is just getting them to your computer. Now I'm going to share this. My computer. I'm going to open up the folder where I copied them. So there are these three files. And what I'm going to do is open up IGB. So in IGB, it's important that, so if it's loaded, you should see this. So you can select which species you want by clicking this little image, or you can go over here to the right under the species scroll bar, go down to Homo sapiens, and then you have to select the genome version. So it's important that you select this second one from the top. This is the HG19 genome. If you just hover the cursor over there, you can see HG19 about 4 lines down. So Feb 2009, that's the genome version we need to select. And it'll take a moment to load that genome. It should look something like this, where it has each gene loaded up. So it automatically loads chromosome 1. And then in the directory where we copied the files over, all you have to simply do is drag these files into the IGB window right here, into this track, and it'll create 3 new tracks. If we hit load data, it'll load the files. The bed file will load fastest. So this is just a simple file. We'll show a line at each significant peak that was called. Then it'll load the wiggle tracks, or the big wig rather, for the input in the stat1 chip. Okay, I'll give everybody a moment to catch up if you need to, or ask a question if you got stuck. And in fact, why don't we take a 5-minute bathroom break? So ask questions, or if you need to take this time to catch up or whatever you want to do, go for it. So I'm going to pause the recording and we'll return in 5 minutes. So something that we still need to do, or at least I do anyway, is do the other peak calling on the 30-minute interferon alpha treated sample. So what I'm going to do is go back to my command, which just by scrolling up, or you can go to the slides. It was on slide, no, we're jumping around, sorry about that. Slide 52, the second command we still need to do, or at least I do. So while that's running, we can play around on the Genome Browser. Okay. So that's going. I'm going to switch back to IGB. So hopefully you guys got your files loaded up. What we're going to do to check our chip is we're going to hit advanced search here. Wait, let me make sure I'm sharing the right screen. Okay. Advanced search, we can type in any gene that we want to look at. So I'm going to type in a gene called IFIT1, which is an interferon-induced gene, interferon-induced protein with tetratricopeptide repeats, I think, 1. And if you click on that, double click, it'll take you to that gene. This is on a different chromosome. This is on chromosome 10, so the data won't automatically be loaded. And what I want to do is it focuses the window on exactly the TSS, the transcription start site, and the end of the 3' UTR. So I want to zoom out a little so I can get more of a picture of the surroundings. And the best way to do that is to grab this sliding zoom bar right here. There's like a little ball in it. If you click and drag to the left, it'll zoom out. You can do it finely. If you just clicked on this, it'll zoom out in more wider steps. So I like to do it like this. So you can see this is a region of the genome where there are a cluster of these IFIT genes. There's IFIT5, IFIT1, IFIT1B. I think there's more to the left. Yeah, IFIT3 and IFIT2 somewhere. So I'm going to hit load data, and our tracks will load. It might look a little odd So what we want to do is we want to adjust the y-axis because this will be set automatically. So if you click on the track on the left where the name is, you can click on both at once if you hold shift. Again, so click, hold shift, click on the other, and then go to graph. Then we can set the minimum Y scale and the maximum Y scale. So for the minimum, I'm going to set it to 0. And for the maximum, I'm going to set it to 40 because I think that's where the highest peak is, just below 40. And once you do that, hit enter, and it'll adjust the tracks. So now both tracks are on the same scale. We are looking at a valid comparison. And for the bed file, again, this is going to demarcate the peaks that were called significant. So it's just a little bar under each peak. And you can adjust which one's on top, which one's on bottom. So right now I have the input on top. If I wanted the input below, you can just drag it like that. And this is, right now it's showing the isoforms for each gene collapsed into one gene track. But if I wanted to see the isoforms expanded, you hit this plus sign for the ref gene part. Okay, and then that'll expand, but I'm not really interested in that. And then, so these are the genes on the plus strand. These are the genes on the minus strand right here. And then it gives the genomic coordinates. So what we see when we look at our STAT1 chip is we see peaks here. See, there's a peak here right at the transcription start site of this IFIT3 gene. There's another peak here, which is actually another TSS for a different isoform of IFIT3. There is no peak at IFIT1b, but there is a peak here at near the TSS of IFIT1 and a peak at the TSS of IFIT5. You can see there's clear enrichments at these sites compared to the input control. And even a little one that was at the TSS of IFIT2, although it wasn't called as significant enrichment, you see like the enrichment wasn't very good and there's not a bar under it where the bed file is. So maybe if we relaxed our criteria for peak calling, this would come up significant, but you can see it's not very highly enriched, at least compared to these peaks. So what else you can do? I recommend if you want to scroll back and forth, you can, of course, use this bar down here, but that will make you scroll really fast and it's kind of hard to control. What I like to do is just hold down a click on the track here and drag the cursor to the left and it'll slide over more finely. So you can, if you don't want to scroll too far. So sometimes if you hit load data, it'll make this big, it'll expand. So if you want to collapse this again, you hit this minus or plus, so it'll collapse the isoforms, make your tracks bigger. And you can push this over to the side to expand the Genome Browser viewing. I'm going to scroll back. So clearly we have STAT1 enrichment at genes that would make sense for STAT1 to be bound near. These are interferon-stimulated genes. STAT1 is binding to the promoters of these interferon-stimulated genes upon interferon alpha treatment. So I will, in case you guys got lost, I will demonstrate all of that again. I'm going to exit IGB. So what I'm going to do is open IGB, which isn't the fastest program, but shouldn't take too long. So, oh, and in case you missed it before, I started running the other max2 peak calling for the 30-minute interferon alpha treatment, which is, the command is the second command found on slide 52. So that's running right now while we're using IGB. Okay, so somebody asked a question about looking at the normalized signal. So is that in terms of, if you mean in terms of, for example, I don't know what happened to this share. For example, in terms of the, so there isn't really a normalized track generated, but if you want to look at the signal after, say, dividing the input from the chip signal, I can show you how to do that in IGB. So I'll show you how to do that in a second. So again, IGB opens. Under species, you need to select Homo sapien, and it's important to have the correct genome version, or else the tracks will be off with the genes. It's February 2009. This is the HG19 reference genome that we're using. So it loaded the reference genome. Again, you can expand isoforms by clicking this plus and minus. I usually have them collapse, especially because I'm using a little laptop and I don't have a lot of screen space. The next thing you do is you want to drag your files into IGB. Another option is to open the files this way, but I tend to like to drag them over because that it's usually easiest, especially if you're dragging multiple files. So to do that, I have the files here in my ChiP-Seq workshop folder. Sometimes it gets a little frozen. I'll try to do that. Something's weird. This is why. Okay. Because I tried to do open file, so it opened up another window. But so you can drag them. Maybe not. I think I broke it. So I'll just do it the other way then. Or you know what? IGB froze. That's why I upset it. It's kind of a temperamental program. Might have to quit it and reopen it. Okay. Still frozen. Okay, I'm gonna have to reopen it. Sorry about that. Okay, finally. So again, species, Homo sapiens, genome version, we did HG19. If you were to do HG38, which is December 2013, you click this one, but what we did was HG19. It's not the newest genome reference, but it's compatible with every program that we want to use. All right, so we have this open. I'm going to gently drag in the two bigwig files in the peaks bed file. All right, so those are grayed. They'll be gray until you hit load data. And for now, I want to only look at the ifit cluster. So if ifit1's in that cluster, I hit advanced search, type in ifit1, and we have the different refseq IDs for ifit1 that pop up. I'm click on the first one. And now I see the ifit1 gene from the 5' UTR to the end of the 3' UTR. I like to move this over, the chromosomes to the right, and increase my view. And when I'm not using this bar down at the bottom, I like to lower that to increase the Genome Browser viewing. Now I'm going to zoom out a little by clicking onto this zoom bar and dragging to the left so I can see this cluster of 5 genes I'm interested in. Hit load data. Now as you can see, it looks really screwy. And the reason why that is because what it automatically loads is the highest Y value. So the Y axis is set to the highest Y value for each track because the input tracks have low counts, not peaks. The Y value gets set to 3, and it looks like I have this weird, artificial peaks throughout this region. But of course, that's just because we're on different Y axes. So if I click these two tracks, pull this back up, hit graph, pull it up a little more, I can see the Y-axis scale. I'm going to set the minimum to 0, and I want to set the maximum to 40. And now I get the nice view of the peaks at the promoters of these genes. Scroll over to the left a little, and every time you scroll to a new part of the genome, you have to hit load data for that to load. And let's say I want to see what the signal looks like if I divide the chip by the input. So a way you can do that is by, I think you select both of these tracks, but we'll see about that. Then hit track operations. Yeah, so you can sum the two signals together. So this would, I don't know why you would do that, but you would add the input signal plus the chip signal. So, whoops. Again, what I did was I selected the tracks. I right-clicked track operations. And what I want to do is do the ratio. Okay, I think that's what it is. Yeah, so what this is this new track that was generated is the division of the chip signal over the input signal. So you can also, let's say you have a normalization factor you want to add. Let's say you want to compare this track to a track that has half as many reads. So kind of a dirty way to normalize would be to do track operations. You can divide this track by 2 if we want to have half as much reads, and then it'll create another track where it's this one up here where the signal was divided by 2. You can see it looks similar, but if you look at the y-axis, it's at 15 instead of, or it's probably at 20 instead of 40. So that makes sense. So those are two things you can do. You can, of course, change the colors if you don't like the default blue. If you right-click, hit Customize, foreground if I want to make it whatever, green. Now that track will become green. If I don't like the label given here, you can also adjust that with Customize and change the track name if I want to call it just stat1. Apply, and now it's just called stat1. And let's say I have an interesting gene that I want to make a screenshot of. So of course you can just do a screenshot using your keyboard, or you can export an image. You go to Save Image, and you can save it to a folder of your choosing, and select if you want the whole frame, which has even the all the buttons and the label. I don't know why you'd want that. The main frame cuts everything out, even the labels, but if you want to keep the labels, you can do the main view with labels. and then you would hit save, and then you have an image that you exported from the Genome Browser. Okay, so that is IGB. And it looks like our chip gave us nice peaks at interferon-stimulated genes, just a few that we wanted to look at. But we want to get a more genome-wide view instead of having to scroll through genes that we're interested in. So we made the files where we will be able to do that. So we'll do some of those tools tomorrow. I think there's just a couple more things we're going to do today that will be relatively quick. Let me go back to PowerPoint, see what we have planned. So we saw something like this when we looked at the Genome Browser. So we want to find differentially bound regions. So we want to use statistics to see between samples which peaks are differentially represented between two different files, maybe two different treatments, like a knockdown or a drug treatment. In our case, it's going to be differences in peaks between a 30-minute interferon alpha treatment and a 6-hour interferon alpha treatment. The differences in stat1 peaks between those two different time points in interferon alpha treatment. So we're gonna, you can also do this similarly with Max, but we're gonna try to diversify our program base by now using a different program called Homer. And Homer has a whole suite of tools, mostly ChiP-Seq tools, but has other assays, other genomic data tools, like such as RNA-seq. But what we're going to use is a particular program called Get Differential Peaks. So what this is going to do is it's going to tell us which peaks are differentially represented between two different files, which is going to be the 6-hour interferon alpha and the 30-minute interferon alpha treatment at the peak locations that we tell it to look at. So these are going to be the peaks that Max told us were significant. So we're going to say at those locations, where are the, which of these peaks are differentially represented between the two different time course treatments. So the output is going to be an Excel file where it's going to tell us for each peak, it's going to tell us the full change between the two files and then a p-value in terms of the differential representation. And it also outputs something called a focus ratio. And basically what that is just, it's a way to measure how focused the peak is, meaning that if we're having the same binding site for an individual peak, you would imagine that each fragment that was aligned in this peak, the reads that align to the plus strand will be found to the left end of the center of this peak, which is the potential binding site. And the reads that align to the right side of this peak, the right end of the fragment should align to the minus strand and should be found to the right of the peak, right? So for example, if it was unfocused binding, if it was binding at regions sort of around the center of the peak, then you'll have a lot of reads aligning to the minus strand that are found to the left of the center of the peak. So this would happen if you have binding sites that are not focused and sort of scattered around in this region. And an explanation for why this could happen is artifacts of cross-linking, where the factor might be binding at the same region in every cell, but it could be cross-linking to neighboring regions artificially. So when you sonicate, you're getting sonication and ends generated that are on the opposite sides of where you would expect them to be found. So this is just, I just wanted to explain what that was, because one of the columns is a focus ratio. And what Homer thinks is a focus peak is if 80% of the reads are found on the end that you would expect if the fragment consists of a focus binding site. And so I'll demonstrate how to use this differential peak calling script. So what we're going to do is we are going to load a program called Homer. Luckily, we don't have to install it. It comes pre-installed on the Hoffman cluster. But what we are going to have to do is generate these tag directories, which are required by the program Homer. So these are generated actually from the BAM files. So using the make tag directory command, you tell it what the name of the tag directory is and then what file to use to make these tag directories. And basically, it's gonna convert the information from the BAM file into file format that's easily used by Homer programs. So it'll make the Homer process quicker when we run the Homer programs. And then we're going to actually run the get differential peaks program. So we're going to do it twice. So first we're going to use the peaks from the stat1 30-minute interferon alpha treatment. So the stat1 30-minute interferon alpha peaks.narrow file, which is essentially a bed file. And we're going to use the reads in the stat1 30-minute interferon alpha treated tag directory that we generated and compare those to the stat1 6-hour interferon alpha data. And then what this is going to do is it's going to tell us which of these peaks are differentially represented for the 30-minute data compared to the 6-hour data. So it's going to output the peaks that are significantly different for the interferon alpha treated peaks, which were called from max2. And then we'll do the same for the 6-hour interferon alpha treated peaks. Which of these peaks are differentially represented for the 6-hour interferon alpha treatment versus the 30-minute interferon alpha treatment? And we're just going to export simple text delimited tab files. All right. So in order to do this, though, we have to make sure our peak calling finished for both of our files. So for me, they both finished. Hopefully they finished for you too. I'm not sure when this finished, but since they did, I will now start with the differential peak calling. So first thing we do is module load Homer. And we are going to make tag directories with the make tag directory command. So first we're going to make a tag directory for the stat1 30-minute interferon alpha treated data. So that's the name of the tag directory that we're going to generate. And the BAM file we're going to use is the one we generated earlier. BAM like this. And it'll take, I don't think it takes too long to create this tag directory file. So as soon as that's done, I'll do the same for the 6-hour treatment. And then we can run the get differential peak calling script. And with these peaks that we are determining as differentially represented between the two different time treatments, you can use those for whatever you want, really. You can see which genes these peaks are associated with. You can see what transcription factors might be binding there by looking at transcription factor motifs. So these are things we'll be doing tomorrow. So after we do this differential peak calling, we'll be done for today. And tomorrow we'll do some more visualization of the data. So there was a question about replicates. So obviously we're not using replicates, but a lot of times you might have replicates in your CHIP data and you don't want to have to just merge the samples. You want to determine differential peak calling between replicates, which is a much more sophisticated process. And while we aren't going to be doing that for this workshop, because we really don't have time to be doing that, plus all the other visualization programs that we're using, a recommended program would be DSeq2 or EdgeR, which both are used with the diffbind program. So a program that's called diffbind, which is an R-based program, would allow you to call differential peaks between among replicates, right? So it'll, yeah, if you have replicates and you want to see which peaks are enriched using more sophisticated statistical analysis, then I wouldn't use Homer. You would have to, I would recommend using diffbind, yeah, which slaves other programs, so it's kind of confusing, but yeah. Hopefully with enough background that you get from this workshop, you'll be able to interpret how to use other programs such as diffbind. All right, so that tag directory finished. Now I'm going to make the 6-hour one. All right, so I've created the tag directories. Now I'm going to, I think we already loaded Homer, so we can run this get differential peaks, which is pretty quick, I believe. Spelled it wrong. Okay, get differential peaks, stat1, 30-minute, -a peaks. So we're telling it where the peaks are by inputting this peak file that was generated from Max. We're saying use the stat1 30-minute interferon alpha treated data that is stored in this tag directory and compare it to this stat1 6-hour interferon alpha treated tag directory. And we're going to output IFNA 30-minute. So this is just an arbitrary name I'm giving the output file, which is the interferon 30-minute diff peaks. Right, now it's testing those locations for differential enrichment. Only 9 of the peaks were found differentially enriched in the 30-minute over the 6-hour. So that's not a whole lot. That means that our data, our peaks are pretty consistent between the two. So let's see when we do it with the 6-hour. So I'm just gonna retype it out. Differential peaks, stat1. This time we're telling it to look at the 6-hour interferon alpha peaks and use the 6-hour interferon alpha tag directory versus the 30-minute interferon alpha tag directory. Output, now I'm calling this IFN a 6-hour diff peaks. Oh, and if your numbers are a little bit different than mine, don't worry. I've noticed that we're getting inconsistent read numbers from the geo download. So actually, it would be, yeah, it's likely that your numbers might be a little different than mine. So now with this, we got more differential peaks, probably because there's more peaks that were called for the 6-hour treatment. So I guess there's probably more stat1 binding with the 6-hour interferon alpha treatment versus the 30-minute. So out of the 931, 263 were represented differentially in the 6-hour versus the 30-minute. All right, so why don't we take a look at what the file actually looks like. I'll look at the 6-hour one. So it has some information at the top. And it tells you the peak ID, the chromosome, the start and end, so the location. Actually will not, so in order to get this, let's see, it's hard because these never align well, but so I think it won't give a score or the focus ratio unless you use the Homer peak calling, but I wanted to use max2 peak calling. So if you're interested in focus ratio, you have to use the Homer for the peak calling. It tells you the total tags for both the the first tag directory and the second, which is considered the background tag directory. And it tells you the fold change versus the background and the p-value. And it's that simple. And now we know which peaks are differentially represented between the 30-minute and 6-hour interferon alpha treatment for STAT1 ChiP. And let's see what else we got. I think that might do it, but let's make sure. So yeah, we looked at that. Yep, so that's it for today. Tomorrow we'll generate even more fun graphs because we already have our all the files we need to do that. And I'll stick around for a couple minutes to let people leave or ask questions if they need to. So yeah. So feel free to leave or hang around to ask questions. And otherwise, I'll see you guys tomorrow. So I'm going to stop the recording now.